Welcome again to Propellicure's video series, where we discuss current advances with leading scientists in Crohn's research. Today we have joining us Dr. Ben Woolston and Justin Hayes from Northeastern University. Dr. Woolston joined the Northeastern University's Chemical Engineering Department as an assistant professor in January 2020. As a National Science Foundation Graduate Research Fellow, Dr. Woolston received his PhD in chemical engineering in 2017 from MIT under the guidance of Professor Greg Stephanopoulos, where his research focused on the development of genetic tools to enable metabolic engineering in anaerobic CO2-fixing microbes. His postdoctoral work was conducted in the laboratory of Professor Emily Balkas at the Chemical Biology Department at Harvard University, where he studied microbial metabolic pathways and enzymes that contribute to the stability of health-associated lactobacilli in the human vaginal microbiota. At Northeastern, his research program, currently funded by the Department of Energy and National Institute of Health, combines approaches from his previous research training in metabolic engineering, synthetic biology, biochemistry, and microbiology to engineer microbes for biofuel, biochemical production, and as diagnostics and therapeutics in the human gut microbiota. His work has been recognized through a range of awards, including the 2020 International Metabolic Engineering Society J. Bailey Young Investigator Award, the 2021 Biotechnology and Bioengineering Daniel I.C. Way Young Investigator Award, and more recently, the NIH National Institute of Biomedical Imaging and Bioengineering Trailblazer Award in 2022. Outside the lab, he is an avid tennis player and a triathlete, completing his first Ironman at Lake Placid in 2018. Pretty impressive. Justin joined Northeastern University Chemical Engineering Department as a National Science Foundation Graduate Research Fellow in 2020. Currently, he is doing his PhD under Dr. Ben Woolston and Dr. Ryan Kopis. His research is focused on leveraging synthetic biology and organ-on-a-chip technology to probe human microbiome interactions. He recently spun out a microbial therapeutics company from his PhD work, Concordance Therapeutics, which is focused on developing microbial therapeutics for intestinal diseases with high unmet needs, such as inflammatory bowel disease. Outside the lab, he is interested in traveling the globe and spending time with friends and family. So welcome, both of you. We look forward to hearing about all of this stuff that you have to offer us. Thank you.
We're so excited to have both of you here. I think in the world of IBD patients and caregivers, we tend to think of researchers as, the GI researchers, immunologists, but we don't automatically think of chemical engineers. And you all have, and others like you, have been doing a lot of exciting work in this field. Before we dive into the specifics of your research, we want to touch on one really exciting tool among others that you're using called organ-on-a-chip technology, or specifically gut-on-a-chip technology. And you all talked about it in a study published last year in Cell Reports, I believe. Can you explain for us exactly what gut-on-a-chip technology is and how scientists use it?
Thanks for the question, Angela. So maybe I can just touch briefly on the first point, why chemical engineers are involved in this space. And then I'll turn it over to Justin, who actually really led the work to talk about the organ on a chip. So when you think of chemical engineers, you think of big oil refineries and smokestacks and these types of things. You don't typically think, right, of biomedical research. But really, I think what we've seen, that this field has expanded in the last 30 years or so. Realizing a lot of the tools that chemical engineers have, a lot of the way we think about complex problems, really apply themselves quite well to understanding really complex biological systems like the human gut, right? We specialize in thinking about complex systems and how they're built from individual components and these types of things. So yeah, as you said, more and more chemical engineers are getting involved in this space. We're learning a lot about the medical aspects as we go. What we think we really bring to the table is kind of that systems level thinking, which I think is really important for diseases like Crohn's disease, which are really complex, right? There's not a single, one bacterial species or one particular genetic factor. It's really this kind of complex ecosystem that leads to it. And so that's why we're hopeful that we can make some contributions here. Justin, do you want to talk a little bit about the gut on chip system?
Yeah, sure. So to give the audience a framework, typically when researchers, scientists are interested in studying the gastrointestinal tract, there's typically two model systems that they will use, right? The first is sort of like the classic petri dish, where you can imagine there are just human intestinal cells in a flat dish. There's some media on top, and they will add different chemicals or different materials to really understand how do human cells respond to different factors within the gut. Now, there are a few problems with this, right? So obviously, in the body, the gastrointestinal tract is very complex, right? There are different cell types. There's not just intestinal epithelial cells, but there's immune cells, there's nervous system cells, there's blood flow. There's also luminal flow, right? So all the food that you eat, all the bacteria that interact with your intestinal lining, there's all of that. So it's a very complex environment. So just like a flat petri dish that you would find in a lab, it's not very representative. Alternatively, people use more complex models, right? Like animal models. So typically mice or rats. The problem with these models, which is quite obvious, is they're not humans, right? So any findings that we glean from these animals, they don't always apply to humans, right? And so the proof is sort of in the pudding here in terms of drug development. I think most people are aware of this, that most drugs that work in animals, when they are then tested in humans, they typically fail, right? Somewhere on the order of 95% of drugs that work in mice don't work in humans. And so they typically fail in humans due to drug toxicity or lack of effectiveness, right? And so that's a pretty scary prospect. What's even worse is that some of the drugs that fail in animals would have actually worked in humans, and those drugs will never make it into humans, and they're shelved and never used. And there's some good examples of that in the scientific literature. So those two model systems are just really not great. And so recently, in the past decade or so, starting out of the lab of Don Ingber over at the Wyss Institute in Cambridge, Massachusetts, they really developed these novel systems where they try to mimic human biology better than a typical petri dish. So one of the major drivers of what's happening in the gastrointestinal tract is that there's constantly flow, there's constantly components in the gastrointestinal tract that are moving around, such as the food that you eat, the bacteria that are there. So the devices that they built, these human organ chips, use human intestinal cells, which is really great because they're human cells and animal cells, and they actually are hooked up to typically a pump, and that pump is constantly flowing nutrients and microbes and media through, and that sort of mimics what's actually happening within your gut, right? You eat something through your mouth, it travels through your gastrointestinal tract, and it leaves out the other end. And so that process is actually really important in cell behavior. So they really showed that this really complex system where you have the interface of human cells and this component of flow really mimics better what is happening in the human body, right? And so these novel systems we thought would be a really good use case for studying hydrogen sulfide and its effects on human cells.
So you would say that the gut on the chip is superior to organoid technology, for instance, that we've read about?
Yeah, so organoids are another interesting innovation. They have their own use cases, right? And so it really depends on the research question that you're asking. I think the great thing about the organ chips is that you can have this flow, right? So you can constantly be flowing materials through, which is similar to what you would find in the body. There are organoid gut on chip complexes, and it gets really complicated, but typically I believe, based on the literature, that the organ chip systems are a bit more complex, and you can interface them with the gut microbiome as well, which is a really important component. And so we thought that the human organ on chip, human gut on chip, was more appropriate for the research question that we're asking.
Do you see the chips replacing mouse models eventually, or what's your thought on that?
Yeah, so as an optimist, I say yes. Practically, we'll see. So it's going to take a lot to make these human intestinal systems more complex, right? You need to get vasculature in there, you need to get immune cells, neuronal cells, right? And in labs, just such as Dr. Abigail and Ryan Koppas, their labs are developing these systems, but they're not quite there yet, right? So maybe in 10 or 20 years, we could phase out animal models, but there may be a place for both in the future.
And you mentioned hydrogen sulfide, which you all have particularly focused on, and your lab has done a lot of work on metabolites and their roles in diseases, of which hydrogen sulfide is one. Let's start from the beginning. What are metabolites?
Sure. So I can take this one. So metabolites are basically small chemical molecules that are either made by bacteria as part of their metabolism, part of their lifestyle, or that are taken up by bacteria to fuel their lifestyle, right? What makes metabolites in the gut really interesting is that there's this complex interplay between what the bacteria are doing, right? They have their own metabolites that they're consuming, that they're producing. But then your own cells, right, your epithelial cells might take some of those bacterial metabolites and they might consume them, right? And so there's kind of an interplay between both of them. Yeah, we're basically here, when we talk about metabolites, we're specifically talking about small molecules, things like glucose, things like butyrate that you might have heard about in the context of the microbiome before.
Okay, and you referenced this earlier, but as chemical engineers, what led you all personally to focus on inflammatory bowel disease in particular, or what interest sparked that, if we may ask?
Yeah, so we have to go for me quite a long way back, but I got really interested in metabolism when I was in college, right? So the idea that basically you can take a series of enzyme catalyzed reactions that cells are doing, and this is the basis of all life, right? This is how cells generate energy to survive. It's how they generate the building blocks they need to make more of themselves, right? So I just became really fascinated by the amazing kind of chemical transformations that cells can do. And so I knew I wanted to study that. So my initial interest was thinking about how do we engineer cells to make the materials and fuels we need for daily life more sustainably, right? So that's kind of what half the lab is doing, thinking about renewable, sustainable biofuels and those kind of things. That's what I spent a lot of my PhD doing. But then also, as I was reading more about these microbes, learning more about them, the same microbes we were studying for making biofuels turn out to be present in the human gut as well. And so I just got really interested in, well, what are they doing there, right? And is this important for host health? And so that's where I then chose to do my postdoc in a lab that was really asking those types of questions, not in IBD, but in terms of the vaginal microbiome, where microbes have been associated with preterm labor, bacterial vaginosis, especially in the developing world. This is a huge kind of recurrent problem without a good solution. And so I kind of learned, how to ask these types of questions, right? How to think about, learning what cells are doing in these complex environments, how to, figure out, break open that black box, if you will, and kind of see what's going on. And then, when I was starting my own lab, I think I was really motivated by, learning a lot about how prevalent IBD is becoming, right? So it's on the rise. I imagine you've talked about this in previous video series, right? As the modern lifestyle seems to be leading to more and more cases of this. And yet still treatment options really aren't that great. And so this seemed, really a place where we might be able to contribute, by our focus on what microbes are doing in the gut, right? Now, so not just knowing who is there in the gut, but by thinking about what are they doing and how this might contribute to disease. It just seems something that would be really nice to be able to contribute to, and kind of a lot of open questions.
Great. So out of all the metabolites, why hydrogen sulfide in particular when it comes to IBD, or what made the focus on that seem to be one you wanted to follow? And can you tell us more about what it is and about its behavior? So just introduce us to hydrogen sulfide.
Sure, yeah. So I'll start, but I'll let Justin take over. So as an academic, one of the things that really excites me is to study things where there's a lot of debate or we don't really know what the answer is. And hydrogen sulfide stuck out to me for that reason, right? So it's been studied actually for quite a long time in the context of gut diseases. And so there's a lot of kind of old literature that says, hydrogen sulfide is associated with inflammation. A lot of studies going back to specific microbes that were known to produce sulfide back in kind of the early days. But there's more recent research that's suggesting that actually this molecule may have some protective properties, right? And so I think any time where there's disagreement is kind of a really interesting area to kind of keep pushing, right? Especially when those disagreements are about the connection to human health, right? Because I think if we could really unravel what's actually going on, it could be pretty powerful. So what is hydrogen sulfide? Well, you may have experienced this. It's the smell associated with rotting eggs. So if you've been around that, you might know what I'm talking about. So it's produced from a few different compounds in the gut, but it looks like one of the primary ones is from protein that you eat. So specific amino acids in proteins like cysteine and methionine have sulfur in them. And it looks like the microbes in the gut are able to convert that sulfur into sulfide, basically releasing this as this not particularly pleasant smelling gas. Yeah.
Okay, does it play a role in immunity in the body, do you think?
Yeah, so there's been some really interesting recent literature looking at less actually in the role of, less in terms of IBD, actually in colorectal cancer. Some really interesting studies looking at how the levels of sulfide contribute to immune cell, T cell activation in cancer cells. So that's really exciting work. It only came out either last year or the year before, so really recent, but it certainly looks like there could be some interplay. And the NIH is actually really interested in following this link more, kind of based on some of these preliminary results.
So is your goal to so it's, from what I understand or read, it's a hard gas to work with. Is that right? When you're trying to do that in your research?
Yeah, Justin, do you want to talk a little bit about this?
Yeah.
You're the one actually doing this.
Yeah, sure. I can give some more background as well on sulfide and its role in IBD. So there's observational data going back to the 1990s. So looking at patients with ulcerative colitis, Crohn's disease, basically just taking fecal samples from those patients and measuring hydrogen sulfide levels. And they tend to find that patients with UC or active UC especially have higher and higher hydrogen sulfide levels. And then when they go in there and they actually investigate which microbes are present, they tend to find more microbes that are fond of producing hydrogen sulfide as a byproduct of their metabolism. And so this sort of got the field interested. And then more recently, in the past decade or so, there's been more and more mechanistic data linking hydrogen sulfide to certain IBD pathologies. So this is done in human cell line models as well as in animals. And so, as Ben mentioned, it's a bit more complicated than that, right? So sulfides sometimes found to be anti-inflammatory in certain cases. And then in other studies, they'll show hydrogen sulfide is pro-inflammatory and leads to cell apoptosis and cell death, right? And so it's sort of this complicated story. And so, yeah, that kind of inspires us to tackle these tough issues and really get down to the nitty gritty and really understand what is the role of hydrogen sulfide, right? And sort of the hypothesis and what sort of inspires this paper is that we think, and we show in this paper, that the concentration, the amount of hydrogen sulfide present in the gut is really dictating its role.
So is your goal to seek to manipulate the levels of hydrogen sulfide in the gut? And if so, how do you go about doing that?
Yeah, so that's a good question. So yes, we aimed in the study to do that very thing, right? So to be able to manipulate the hydrogen sulfide levels within the gut. And so, as I mentioned previously, typically what researchers have done in the past is they will dose in the chemical forms of hydrogen sulfide. So they'll put them in a petri dish or in a mouse. Now, the problem is, as I mentioned previously, is that the hydrogen sulfide will either evaporate or react very quickly. And so then it's really difficult to study how does hydrogen sulfide, the compound, affect the human cells or the animal, right? Because it's going to evaporate and react. So it's actually very difficult to work with. So what we did in this study was, first, the two real innovations here are the organ chip, the gut-on chip that we are dealing with, is designed with a material that actually maintains the hydrogen sulfide within the device, so it will not allow it to evaporate out. So now we can actually study how it affects the human cells. And so then we engineered the bacteria to be able to produce small amounts of hydrogen sulfide so we can investigate, okay, what is the role of low levels of hydrogen sulfide on human biology? And then we also engineered our bacteria to produce a lot of hydrogen sulfide and then really begin to understand how do high levels affect human biology. And so we can get into the how do we do the engineering of the microbes if you're interested.
Yeah, I mean, that's so you all are engineering your own bacteria to put into the body and modulate those levels of hydrogen sulfide, it sounds like. Is that right?
Yeah, that's exactly right. So just kind of adding to what Justin said, I think one of the things that's really powerful about this is it really mimics what's actually happening in the body, right? When you think about microbes producing sulfide in your gastrointestinal tract, right, that's not happening all at once everywhere because of an injection, right? That's happening precisely where the bacteria are interacting with the human cells in the mucus. And so, as well as the other problems that we have with kind of injecting sulfide or using chemical donors that Justin already mentioned, the other problem is that doesn't really represent how it's actually being produced in the gut. And so, the idea was, well, over the last 10 years, the field of synthetic biology and metabolic engineering that we work in, this is where our expertise in. Basically, we've got really good at engineering microbes to very precisely produce different levels of various compounds, right? No one had really looked at hydrogen sulfide before because most of the time people don't really want to make it, right? We typically think about making, biomaterials and those types of things. But we said, look, we can use the tools that we've got to now, make these compounds on demand precisely in the microbiome to really reflect what's happening natively, right? And so what Justin spent a lot of time doing it really nicely, right, was developing a variety of different strains, all of which could produce different levels, right? And so what's really powerful about this is when you think about now running those experiments, right? You can, the only thing that changes between the different strains is the amount of hydrogen sulfide that's produced, right? So it's really a nice controlled experiment. When people have done this in the past, what they've tried to do is say, well, okay, I'm going to find some microbe that we know produces sulfide, right? And then compare this to the case with no microbe, right? But that's, the problem there is that whatever microbe you choose likely has a bunch of other effects that may not actually be related to the sulfide specifically. Because the way we're engineering this, we're only changing that one specific pathway, we can really be sure that any effect we're seeing is specifically because of this metabolite. And so I'm really excited about the work we've done with hydrogen sulfide. But I think more broadly, you could actually think about using this type of approach to study a number of different molecules that people think are interesting in these types of diseases.
That is very exciting. So how within the environment of the gut and its complex and dynamic nature, now this sounds like this has therapeutic potential. So how did these engineered bacteria survive? Because we always hear about how very little can survive down there because of the environment. So tell us more.
Yeah, this is a real challenge, and one of the things that Justin is really thinking a lot about now is that, often when you engineer a microbe, it becomes less fit. It doesn't survive nearly as well as a native microbe. So we've started thinking a lot about how do we make sure that our microbes will stick around long enough to do what they need to do. But also on the flip side, we have to think about, well, how do we get rid of them at the end, right? Because we don't want to take over the whole microbiome as well. So the delivery aspect that you mentioned, yeah, this is a big challenge. Fortunately, there's a lot of people working on really interesting kind of delivery mechanisms, capsules you can put around bacteria that degrade in the stomach. So by the time it's gone through the stomach, now the bacteria are kind of free and out there. A lot of people thinking really hard about how do we make sure that once the bacteria come out the other end, they don't persist in the environment. So these are all really important things. The nice thing is there are some really smart people out there working on. One of the things Justin is tackling now is, as you mentioned, it's a complex dynamic environment. How do we make sure that whatever precursor molecules we're using that the cells are converting to sulfide, are those actually going to be bioavailable in the gut? And this is part of what Justin is thinking about now, is how do we make the bacteria we've made really robust, right? So they can actually compete in that environment.
Let me just throw out this question, and for the sake of asking, but do you think that the hydrogen sulfide levels might change in a Crohn's patient, like during periods of flares or active inflammation versus remission? Is it kind of fluid like that, do you think?
Yeah, I think it is. I think even in healthy patients, yes. So in patients with a lot of inflammation, there can be a lot of oxygen present, and so that can give rise to certain microbes in the gut that are capable of producing more and more hydrogen sulfide. So the potential link there as well.
So I have a question.
I'm sorry.
Do you feel like, do you believe that this is not really the cause, but one of the main causes of Crohn's, that it's something like this metabolite is creating this havoc down there because of it?
I think it's too early to really say that. What we really don't know from a lot of these kind of association studies that have done before is sulfide the cause, or is it a consequence, right? You could imagine what you just described, right? That the production of this molecule leads to inflammation, and that's a trigger. But you could also imagine that inflammation happens for some other reason, right? Genetic, environmental, the various things we know affect IBD. And what we're actually seeing when we see high levels of sulfide is the bacteria's reaction to that, right? So I think I would be really hesitant to say at this point that it's causal. There's some interesting data kind of in both ways, but that's kind of precisely why we're building these tools, is to be able to ask that question, right? So some of the studies that we want to do, now that we have the strains, now we've demonstrated they can work in these chip systems, it would be great to go into a mouse and say, okay, if I do nothing else other than introduce this microbe that produces sulfide, do we see inflammation, right? Because I think that would be really powerful data toward that hypothesis that you put out there. But I think until we have those really good mechanistic studies that go beyond just association, that actually look at, what is the link between sulfide and inflammation and these types of things, I think it'll be a bit difficult to really say that at this point.
To build.
Go ahead.
Yeah, to just build on what Ben said in my previous comment. It's very easy to establish these correlative links, right? But it really is difficult to prove causality. And so that's really important to keep in the forefront of your mind, right? Just because two things are correlated does not mean they're causal. So that is something that our current research and future research is focusing on parsing out what is cause and effect.
Regardless of whether it's causal, is it possible that just still have that therapeutic effect to address the dysbiosis present in Crohn's and perhaps help make patients better, even if it's not the cause? Does that make sense?
Mm-hmm. Okay, I think that's one of our strongest hypotheses, right? Is if this is a response to inflammation, you can imagine that that's a response that helps to become inflammatory, in which case we want to keep that going. Or if it's a response that worsens it, perhaps correcting that can help to reduce inflammation. Yeah, so I think you're absolutely right. It doesn't necessarily need to be causal to still be a lever that we can pull kind of therapeutically to help out.
And have you thought about during the course of research, perhaps just brainstorming whether it could even, this modulation in the lab could potentially offer a preventative approach, even delaying or preventing symptoms from the onset of symptoms, basically in Crohn's?
Yeah, I think so. I think one of the really exciting things about these engineered microbes is, as well as engineering them to make specific amounts, we can also engineer them to be responsive, right? To actually sense their environment and then make decisions based on what they're sensing, right? So you could imagine that if you could engineer microbes to sense inflammation and you knew that then there was then some specific response you wanted the microbe to do to reduce inflammation, you could target it specifically to inflamed tissue and not worry about it having a response elsewhere in the body. And in fact, researchers have started to do this, right? There are signals associated with inflammation, and people have hooked them up to allow bacteria to sense them and then make decisions. So yeah, I think you could use that for kind of precision. As we know with Crohn's, right, it's not uniform through the gastrointestinal tract, so it would be really nice to be able to target the response to specific tissue. But I think you could do this temporally too. You could, or something that happens over time. You could imagine taking this some sort of bacterial pill that now is going to reside in your gut, and it's just going to be dormant, right, until it detects inflammation, and then it will kind of turn on this response, or even just produce some kind of signal that you can detect to know, perhaps as an early diagnostic of inflammation, right, before a biopsy would be done, to get more information earlier. So I think there's some really exciting potential for exactly what you're talking about.
So would that be done as well for cancer? Or is it just, wow, that's really...
Yes. So people actually, for cancer, this has been done. There was a study that came out of MIT, 7 or 8 years ago now, where from the Bhatia lab, they basically engineered a bacteria, and I may not remember all the details here, so I apologize, but basically this bacteria localized to tumor tissue, and then produced a signal that could be detected in urine, right? So you could actually detect fairly early on small cancerous tissue just based on the fact that this particular bacterium kind of was naturally attracted to colonize the tumors. Yeah.
Interesting, because I was going to ask what other diseases, well, we talked about colorectal cancer earlier, besides IBD could be targeted with this approach, and then you can apply the lessons learned to any other illnesses or conditions or diseases besides cancer and IBD that you all have looked at.
Those are the big ones that come to my mind, and I think the reason that those have been kind of good first targets is because the environment in those diseases, right? So in the gastrointestinal tract and in tumors tends to be a low oxygen environment. And so it tends to enrich for certain types of microbes. And so because of that, you can use that fact to kind of target your microbe to that specific area. And so there's actually, there are reports of using bacterial infection to cure cancer back as far as the 1800s, which is kind of amazing. So yeah, I think to me, those are kind of the two biggest areas that people are excited about right now. But yeah, I could imagine there might be other types of diseases too.
That's amazing. And do you see, what about this approach as maybe a point of care diagnostic tool even, that one could assess one's patient's levels of hydrogen sulfide and truly have precision medicine in that sense?
Mm-hmm.
Do you think? Yeah.
Yeah, absolutely. So one other aspect of the work that another student is really working on is engineering the specific bacteria to sense what is the level of sulfide and then produce some kind of measured signal as a response to that. And it's exactly as you're suggesting. You can imagine using that as an early diagnostic, or even just to get better data than we currently can on what are sulfide levels, right? I think part of the reason there's so much confusion in the literature about is sulfide helpful or harmful, is it associated or not, is because just getting accurate measurements is so difficult, right? And so I think having a tool like ours where you could imagine actually, visualizing the whole length of the gastrointestinal tract and getting kind of a map of what the local sulfide levels look like, that would be so powerful for all of these types of things.
I have a hard time imagining how these bacteria are able to zone in on areas where there would be inflammation. I mean, it's just like you put a coat. I mean, how does that work?
Yeah, great question. So a lot of it, again, comes down to metabolites, right? So there are specific metabolites that tend to be found in inflammation. And so if you can program your microbe to respond to that metabolite, essentially then it's going to respond to inflammation. And so some of the earliest kind of detections of sulfur-related compounds in IBD was exactly doing this. So there's a molecule called tetracyanate, which is produced actually buy human cells from the hydrogen sulfide that bacteria produce, but it's only produced in a oxygen heavy inflamed environment, right? And so people use that as essentially a proxy for inflammation that bacteria could be engineered to detect. Yeah,
I see. Okay.
Wow. Do you see engineered microbes then as being the future of microbiome-based therapy? And how does it compare maybe to FMT or fecal microbiota transplantation when healthy human donor stool is transplanted from one person's gut to another? I mean, what are your thoughts on that?
yeah. Justin, do you want to take that one?
Yeah, sure. So I think that FMT is probably generation one of the therapeutics for the microbiome, right? Currently, as we've mentioned a lot here, most of our understanding of the microbiome is all correlative, right? A lot of associations. We don't really know causality, right? And so the promise of FMT is that it's consortia based, right? It is an entire microbiome from another human, and so we don't really need to know exactly how it works as long as it works, right? And so that's a beautiful thing. And there have been 2 therapies that have been approved in the past year for C. difficile, so that's exciting and promising. I think as we continue our research, as other people continue their research, begin to understand causality in the gut, right? We can hopefully then make the generation 2 therapeutics that are very targeted, and we understand very in detail the mechanism of what's going on, and we can produce better therapeutics. And that's sort of the thesis of my work in the company that we spun out.
So thank you for investigating this area. What do you see as the next steps of the research? Can you tell us a little more in detail of what you're doing right now?
Yeah, I can touch on it briefly a bit. So Ben has mentioned this a few times already, but we're interested in going into animal models now, and we're interested in understanding the pro or anti-inflammatory role of hydrogen sulfide. So we'll use various IBD disease models to really investigate, does sulfide actually exacerbate disease or make disease better? And independent of disease in healthy mice, does it actually promote inflammation, or is it an anti-inflammatory? So really next step is to really use the engineered bacteria for this first study, and now apply them in animals to really begin to get at the mechanistic understanding of hydrogen sulfide and its effects on mammalian biology.
And you all alluded to earlier your spin-off company called Concordance Therapeutics. Tell us more about that and how that happened, and what you're doing with that and how that works.
Yeah, so sort of already touched on it briefly, but we believe that as we begin to understand the mechanism of how different metabolites affect human cell behavior and human disease, that we now are developing therapeutics to go after some of these diseases, right? So Ben has touched on it a bit previously, is can we build these sense and respond bacteria, use them as a platform to really dynamically regulate metabolite levels in the intestines for different disease pathologies? So hydrogen sulfide is one metabolite, right? And if we understand its role mechanistically, we'll be able to modify its levels in diseased patients and hopefully be able to resolve certain symptomologies or clinical biomarkers. And so that's sort of the thesis of the spin-out company. We're making more and more progress in that direction, and so we're excited to see where it goes.
So wonderful. What have we not touched on that might be interesting for our viewers to know of your work or what you're doing or have done? Does anything come to mind?
Great question. I think I would bring it back to, I think the microbiome area has become really exciting and really exploded in the last few years, right? And I think that research has done a fantastic job of asking the question, who is there, right? What microbes are present with disease, without disease? But I think what really has to happen if we really want to translate the microbiome, right, and actually use this for generating new therapeutics, new knowledge about these diseases, we have to go beyond just who is there, but also really ask, well, what are they doing, right? And that's hard, right? The reason for that, we're getting really good at sequencing the microbiome, right? So it's very quick now. If I wanted to get all the genes from all the microbes in my own microbiome, it would be pretty straightforward to do. So you might think, well, with all that information, we should be able to predict what they're doing, right? Well, it turns out about 30% of those genes, we have no idea what they're doing, right? So, I think that's kind of the forefront of this realm right now is to, figure out what is the function of these microbes and how do those functions really interact with host cells and what the host is doing to lead to these diseases. So yeah, we're really hopeful that the platforms, the tools we're developing will help contribute to that. They're certainly not the only way of doing that, but I think that's what I'm really excited to see over the next 10 years or so, right? Kind of really delving into what's happening in the microbiome, right? What are the bugs doing, and how does this really affect us? Because what we're starting to know, right, is let's take IBD for example. There's not one specific microbe that causes IBD, right? It's probably a combination of genetic factors, environmental factors, microbiome factors that together take a microbe that normally is fine, right? And can hang out in the gut and have no problems. But all of a sudden it's presented with an opportunity. It's doing something it wouldn't normally doing. It's in a strange environment. And that's where we start to see the inflammation. So I think really digging into the mechanisms behind that is really going to be, I think, transformative for all these kind of bacteria-related diseases. And so it's really exciting to see the tools are getting better and better, right? They're doing the science, are getting cheaper and cheaper. Seeing platforms like Justin has developed with the gut on a chip, this now gives us kind of unparalleled ways to look in real time, right? We can actually observe under the microscope what the cells are doing in these chip models, right? So I think all of this is just, to me, is just a really exciting time to be in this field and to be asking these questions.
I'm sorry, Angela.
No, I was just going to say that makes perfect sense, and we're so appreciative of you all and the work others are doing in this. Annabelle?
Yeah, I'm curious about the timeline. I mean, you mentioned 10 years in terms of trying to figure out like what's going on. How about like in terms of, first of all, like the gut on the chip? Now this to me is very interesting. When I first started getting, when my son was first diagnosed, and you start looking at possible what's going on and so-called cures and things like that, and then you would hear about, was this particular treatment cured the disease in mice? And then I remember a woman who was doing research just looked at me and she goes, "Do you know how many mice have been cured of cancer?" And so from that point on, I've been very cautious about studies based on mice. So this gut on a chip is is just wonderful to hear, just like organoids. I mean, I really like that idea as well. And so what's the timeline? Do you think that people are going to start adopting this sort of thing in their research and using this? And so there's that question. And then the other thing that I'd like to know is in terms of therapy with this particular type of research that you're doing, when do you actually see something available for people?
Yeah, so I'll start with the chip question. I do think we're going to see more and more of this in the literature. One of the things that's driving this, just to be clear, is my lab has really done nothing with the chip. That's our collaborators, Ryan and Abby Koppers. They have done a phenomenal job of designing these chips, not only to be really good for hydrogen sulfide in terms of permeability and those kind of things, but also cheap. Right, so they can make their chips. I think, Justin, it's about $3 a chip now, right? The original gut-on-chip systems were kind of hundreds of dollars, so they really weren't accessible to the broader community. And what Ryan and Abby have done is really, I think, transform that. It's going to be much easier for people to kind of scale this up, right, and do these kind of complex studies. Here's the challenge, though. As Justin told you, one of the things that we're about to start doing is mouse experiments, right? Which is kind of counter to what we've been talking about with these chip systems. The reason for that ultimately is that regulatory bodies and funding agencies move really slowly, right? They have to be cautious, right? But so when we go to the NIH and say, hey, we've got this fantastic chip data, right? And it's telling us all about what sulfide is doing. We know from experience that if we don't do the same experiments in mice, the people reviewing the grants are going to say, well, we don't know how to interpret this, right? And it's the same thing for companies, I think, that are kind of developing drugs, right? There is no FDA pipeline based on chip systems yet, right? It's set up for mouse studies and canine studies and these kind of things. And I think there's good reason that these things should start to shift, but that just doesn't happen quickly with these agencies, right? They're highly risk averse for good reason. They slow bureaucratic turnings. So I think that's one of the things that's kind of slowing things down. But as I say, as these get cheaper and you see more and more basic research happening with these systems, I think people will start to kind of pick them up more and more. In terms of when do I think therapeutics are going to come out, I really don't want to give a number answer on this. We'd like it to be as soon as possible. And really what motivates us in this field is, how do we improve lives of patients now, right? So we always have that in mind. It would be really hard to say, at least from my perspective, how long that's going to take.
Any other questions? Well, we would like to thank again Professor Ben Woolston and Justin Hayes from the Woolston Lab at Northeastern University. Thank you so much for the pioneering work you're doing. I'm Angela with Propel a Cure. Thank you for joining us.